commercially already used for Alzheimer's disease. So I thought that liposomes and polymeric nanoparticles, these are all a type of polymeric nanoparticles. So among these solid lipid nanoparticles, I had chosen my work, and among these solid lipid nanoparticles, these two fish oils, as they are having rich fatty acids, I had chosen this, and I prepared this. Our team prepared these solid lipid nanoparticles and loaded with the drug Rivastigmine, and we had seen certain characterization studies, whether they are sustainable and they are showing. So this is my topic: Rivastigmine loaded solid lipid nanoparticles, formulation, characterization, and in vitro antioxidant activity. So these are the contents: introduction, experimental details, results and discussions, and conclusions. So objective of the present study is to develop bioavailable SLNs of Rivastigmine, which would reduce the side effects of conventional formulation and enhance penetration of drug into the brain, increasing the treatment efficiency with sustained release action. This is plan of work. Formulation development. So for this, selection of materials, in that selection of lipid and selection of polymer are essential, and formulation technique our team used is melting emulsification, high shear homogenization technique, and then optimization of drug lipid ratio, and then optimization of processing parameters and in vitro antioxidant activity. So Alzheimer's disease, this is the most common neurodegenerative diseases worldwide. Despite all the efforts made by the scientific community, current available treatments have limited effectiveness without halting the progression of the disease. So that is why new molecules such as growth factors, antioxidants have been raised as new therapeutic approaches. However, these molecules have difficulties to cross the blood-brain barrier, limiting its therapeutic effect. The development of nano drug delivery systems may permit a targeted and sustained release of treatment, offering a novel strategy to treat these neurodegenerative diseases. And we all know that nanoparticles have been widely studied in biomedical and biotechnological applications, particularly in drug delivery systems for drug targeting, since their particle size ranges from 10 to 1000 nanometers, which is acceptable for intravenous injection. So these are different types of nanoparticles which are used for drug delivery. So solid lipid nanoparticles are at the forefront of the rapidly developing field of nanotechnology, with several potential applications in drug delivery and research. Due to their unique size-dependent properties, lipid particles offer possibility to develop new therapeutics. Hence, solid lipid nanoparticles hold great promise for reaching the goal of controlled and site-specific drug delivery, and hence attracted wide attention of researchers. The solid lipid nanoparticles are submicron colloidal carriers, which are composed of physiological lipid dispersed in water or in an aqueous surfactant solution. SLNs consists of macromolecular materials in which drug is dissolved, entrapped, and or to which the drug is adsorbed or attached. So advantages of SLNs is controlled and targeted drug release, and excellent biocompatibility, improved stability of pharmaceuticals, high and enhanced drug content, enhanced bioavailability of entrapped bioactive compounds, chemical protection of liable incorporated compounds, no special solvent required, very high long-term stability. So this is the Rivastigmine drug, which is an anticholinesterase agent having a favorable efficacy and safety in patients with Alzheimer type dementia, and has been widely used for the treatment of Alzheimer's disease. It inhibits acetylcholinesterase, hence have additional clinical benefits in patients with dementia related to Alzheimer's disease and recently in Parkinson's disease also. Hence, it was chosen as the drug candidate in the present work, which was designed to overcome the problems of conventional dosage forms and can be used for brain targeting. This is experimental detail. So the material, the drug I had chosen is Rivastigmine tartrate, which is a commercially available one, and fish and flaxseed oil. So as surfactant, I had taken polysorbate 80, and this is the normal method, drug in melted lipid. So drug which is melted in lipid by using emulsification and high shear homogenization technique was used, and then after that solidification, and then that was taken for this is the pictorial representation of surfactant coated solid lipid nanoparticle. So solid core, and then a drug you can see clearly. So this is the drug, and this is lipid core, and then surfactant layer is outside. So this is the Rivastigmine, which is loaded in solid lipid nanoparticle, and surfactant is outside the lipid layer. Then these are the characterization techniques. And so for morphological studies, we had gone for SEM and AFM, and functional groups. For the identification of functional groups, we had taken this FTIR, and optical properties were studied by using UV-vis spectrophotometry. And then Rivastigmine loaded nanoparticles were characterized. Physicochemical properties of Rivastigmine loaded SLNs were studied by using DSE, DLS, and zeta potential. And drug entrapment efficiency was studied, and drug release was also studied. And in vitro, finally, in vitro antioxidant activity was studied by DPPH and reducing power assays. So coming to the results, for the formulation, 3 concentrations of drug was chosen, that is 0.5 mg and 1 mg and 1.5 mg. And that lipid matrix, I told already that fish oil and flaxseed oil were taken, and different concentrations like 1 ml, 2 ml and 3 ml were chosen. And then as the lipid concentration increases from 1 ml to 3 ml, the surfactant concentration obviously decreases, which leads to the increased particle size that we can see in this figure. So this is the mean particle size. So this is the concentration of the drug, and this is the lipid concentration. So as the lipid concentration increased, the particle size is also increased. And then all formulations were prepared with 1% surfactant mixture of polysorbate 80. This is coating. This is used for coating of lipid. So this is the preliminary confirmation of solid lipid nanoparticle formation by using phase contrast microscopy. And these are the SEM images. So this is these are so we taken for all the formulations SEM images. The best ones we have selected from that SLN1 and SLN9. So results obtained by some studies indicates the size of the SLNs influenced by both the oils, flaxseed oil and fish oil. So there is an increase in average diameter of SLNs as lipid concentration was increased from 77.45 nanometers with 1 ml of lipid and 0.5 mg drug of SLN1, and up to 174.2 with 3 ml of lipid and 1.5 mg of drug of SLN1. So increase in size of the particle may be due to high lipid concentration, which cannot accommodate large amounts of drug, that leads to adsorption of RT on surface coated with surfactant. So this is PCS, particle size photon correlation spectroscopy analysis. This showed the average particle size and polydispersity index of the prepared nanoparticles were 248 nanometers. So this is here 248 nanometers and this 0.236 respectively. So determination of electrokinetic potential of SLNs, which is nothing but zeta potential. So the surface particle charge is quantified as zeta potential, which refers to the electrophoretic mobility of the particles in an electric field. Absolute zeta potential of prepared nanoparticles ranges from minus 11.1 for SLN1s, minus 21.1 for SLN6 and minus 16.4 for SLN9. So these close values indicate similar interfacial properties of all developed SLNs and good physical stability in time without further aggregation. From the results, it was observed that the SLNs containing 1 mg of RT, this is about SLN9, have increased stability as compared to other formulations containing 0.5 and 1.5 mg of RT. So these are the zeta potential values. Then this is the images of AFM for SLN1 and SLN9. So RT loaded SLNs were spherical. So AFM images of both SLN1 and SLN9 were spherical and subspherical in shape. The morphology of nanoparticles for formulation of both these were studied. So the topographic images showed a smooth surface without any noticeable pinholes or cracks. All particles were spherical in shape and separated from each other, suggesting possible stabilization of the SLNs due to the presence of polysorbate 80. So coming to the FTIR spectra, functional groups of both RT and RT loaded SLNs were studied, and they had shown different types of peaks at 3398 stretch and also 2928 centimeter inverse. So all these peaks were observed both in RT as well as RT loaded nanoparticles, except the peak. So this is the peak which appeared at 1744, and this indicates the presence of esters. So coming to the differential scanning calorimetry was carried out for investigating the crystalline property and to scrutinize for any possible interaction between the drug and polymer in nanoparticles. So this is one of the most general methods to find out the physicochemical interaction between drug and polymer in formulation. So thermograms. So DSC has been employed in this study to provide the melting behavior and also crystalline structure. So thermogram of RT is showing an endothermic peak at 146.8 degrees centigrade and 2. So this was 146.8. This is for RT, only RT loaded particle, only RT DSC endothermic peak, and these two are for fish oil and as well as flaxseed oil. So thermogram of RT is showing an endothermic peak at 146.8 degrees centigrade and 2 endothermic peaks at 110.5 degrees centigrade and also another peak at 160.24 degrees centigrade. So endothermic peak of RT was completely absent in 146.8 degrees centigrade of RT loaded, which is observed in RT endotherm peak that was completely absent in RT loaded SLNs, which indicates that RT was completely solubilized inside the lipid matrix of SLN. So that peak appeared at 143.94 degrees centigrade for drug and lipids, at 139.32 as well as 186.27 degrees centigrade. This indicates that drug was not in crystalline state in the formulation. So this is a RT loaded SLN's endothermic peak. So coming to the entrapment efficiency, the determination of entrapment efficiency is an essential stage in order to predict the suitability of SLNs based on omega-3 fatty acids as efficient reservoirs for the RT. Entrapment efficiency inside the lipid core was strongly associated with the lipid concentration. So the entrapment efficiency was improved with lipid matrix, which has been increased to a maximum value of 3 ml, that is for 68% for SLNs prepared with a 1 ml lipid and 89% for SLN3, which is prepared with a 3 ml of lipid. But as the concentration of drug increased to 1.5 mg in 3 ml of lipid, as it causes the lipid saturation, so that it cannot accommodate the drug inside the core, and drug gets accumulated on the surfactant that leads to immediate drug release. So these results could be justified by the high solubility of RT into the complex lipid core, which contains the liquid fish and flaxseed oil reservoirs and with a sustainable drug concentration. So this is entrapment efficiency is calculated by using the formula mass of a drug in nanoparticle by mass of the drug used in formation into 100. Then this is the in vitro release study. So the release profiles of RT both as well as RT loaded SLNs was studied. So for a period of 48 hours, the experiment have been compared with the reference RT. So by comparing the release profile of both kinds of RT loaded SLNs and only RT, the release of RT from SLNs was significantly better than the RT alone. After 5 hours of diffusion experiments, RT release has been almost reached to 60% and ended with 76% release at 48 hours. So, but in case of SLNs, there is a controlled release of a drug and reached to maximum release rate of 96% at 48 hours observed with SLN9. The in vitro release behavior of SLN9 loaded with concentration of 1.5 mg of RT demonstrated in the presence of RT outside the lipid core. Certain amount of RT was released from the SLN9 immediately after the release experiment. This percentage obtained during the first 5 hours of measurements corresponds to the free RT, so which manifests a faster release rate as compared to that of RT entrapped inside the lipid core. So this is the diagram that shows only RT, which is compared with different SLNs, SLN taking from SLN1 to 9. So this is antioxidant, in vitro antioxidant studies by using DPPH activity. So all had shown compared to RT alone, the only SLNs, RT loaded SLN showed better antioxidant activity. So this is the reducing power assay. Then coming to the conclusions, various RT-loaded SLNs were successfully prepared and then their physicochemical properties were investigated. The in vitro characterization of RT-SLNs has shown that the lipid matrix plays an important role in improving the antioxidant capacity with a potential scavenging of oxygen-free radicals generated in the DPPH as well as reducing power assay because of the omega-rich fatty acids. So entrapment efficiency and drug release profile depends on the concentration of lipid and as well as drug mixture employed. Then most importantly, this comparative study is associated with the main feature of prepared drug-loaded nanoparticles to exhibit better in vitro sustained release of RT as compared to the individual or RT alone. So these data collectively support that SLN as promising sustained delivery systems better than the immediate release of drugs such as RT. So with this, we have completed preliminary studies, and then we would like to see in vivo studies by seeing this antioxidant activity by taking certain enzymes like SOD catalase and lipid peroxidation that we would like to check, and then after that we'll see whether the particles are reaching to the brain regions exactly or not. This is our future work. Thank you for your patient listening.